Hello, good morning, good evening, good afternoon, depending on where in the world you are. Welcome to this webinar on immunometabolism and its relationship to COVID-19. I am very honored and privileged to have three absolutely stellar people in the group in the webinar today. Dr. Vikas Agarwal, Dr. Durga Prasanna Misra, and Dr. Latika Gupta. And I will ask each of you to introduce yourselves. So Dr. Agarwal.
Yeah, I'm Dr. Vikas Agarwal, professor in Department of Clinical Immunology and Rheumatology at Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow in India. So I think that is enough.
Thank you, Dr. Agarwal. So Dr. Gupta, I think you are next in the alphabetical order.
Yeah, I'm an assistant professor of clinical immunology and rheumatology at SGPJI in Lucknow.
Thank you. Dr. Mishra?
Hello, good evening. I am Durga Prasanna Misra. I am an assistant professor of clinical immunology rheumatology at Sanjay Gandhi PGI Lucknow.
Thank you. Actually, what none of them are telling you is that all of them have many, peer-reviewed articles in peer-reviewed journals. They all lead journals. They lead research. So the wonderful thing about all three of them are they are actual clinical researchers. So they see patients as well as being involved in research. So I'm going to start with the PowerPoint, and I'll just let's hope I can get everything working well. Okay. Can you all see my screen?
Yes, please.
Okay.
Yes.
So for the participants, now this is a webinar format, so you will not be able to turn on your video. But at the end, we will have questions, so you can type either in the Q&A box or we also have a chat facility available. So this is about all of us. So what are we going to talk about today, basically? So this is just an overview because immunometabolism is, I was introduced to this, entire area by a person called Gabor Ordosi. So we are a part of a big Facebook group called the Lower Insulin Group. And I actually am very indebted to that group to help me understand, biochemistry and all the basic sciences, because I am a functional medicine doctor and I'm a clinician. So basic biochemistry kind of is something that I had finished a long time ago. But this is basically what we'll go over is what is immunometabolism, and most importantly, what is its relationship to COVID-19. And the focus will be on glucose and lipids. Of course, there are a lot of other aspects to immunometabolism as well. Then mitochondria, and where does the ACE2 receptor come into the picture, because people have become very familiar with the ACE2 thanks to the ongoing pandemic. What is the role that insulin has? And this is the insulin that is made by the body, the pancreatic insulin, or as well as the insulin that is exogenously administered for treatment of hypoglycemia. Then as functional medicine doctor, so my area is nutrition and food as medicine and supplementation. So for us, just as in the pharma world, people are looking at drug targets and looking at drugs that have already been approved in other conditions. So can we repurpose those drugs for the SARS CoV-2 infection? Likewise, in my world, we look at, are there any nutrition targets? Are there any supplement targets? Things that have already been used before. And in some ways, our job is a little easier because number one, as functional medicine doctors, most of us are not dealing with acute problems. And I know all of you were at the COVID front line. So I am not at the COVID front line. And on the other hand, because the things we manipulate are sort of, people think they're relatively easier because they involve food and nutrition versus, some of the drugs which may have potentially toxic side effects. So quite often I say, my job is easier than, many of yours. I don't have to make big decisions like, is this medicine going to kill my patient or is this going to help them? You all have to make much more difficult decisions. So going by that, does the substrate availability of glucose, proteins, fatty acids, does it change the immune response? So in my world, that's a very important question. And we, of course, don't have all the answers, particularly in relation to the ongoing pandemic, because it's still very new and it's just about a year old and a lot of research still has to come up. And the other interesting question, this is what has been coming up to me of late, is do we need different strategies at different stages of the disease? We know that there are asymptomatic people who can transmit the disease. Then we also know at the other end of the spectrum are the long COVID, people who've gone through an infection already, but they still have ongoing lingering symptoms. That is something, again, not completely unknown from the viral infection world, but at the same time, a lot of people were taken by surprise. And a lot of doctors actually, unfortunately, dismissed many of these patients with long COVID, telling them it's all in your head. But then it turns out that long COVID does exist. And that is probably something we'll have to deal with for a long time. And all of you are very, very familiar in the most of your practices. You take care of patients who have ongoing chronic disease. So it's not like they just show up at the, at the hospital once and then they're cured and gone. So your perspective is very, very important and very interesting because it comes from a very different field compared to most of the other doctors who are in regular clinical practice. So I will hand over now so Dr. Agarwal, are you going to start with this of what is immunometabolism? When did this entire subject start? And most importantly, what is it that for a non-immunologist, how would you explain something like this? Because this is like going back to biochemistry in undergraduate days, and I don't think I was paying much attention then.
Yeah, none of us were actually, except passing the exam.
That's good to know.
Yeah. So Dr. Durga will actually like to give a perspective on the
Okay, Dr.
immunometabolism.
Durga, over to you.
Actually, this immunometabolism initially came into work with respect to cancer chemotherapy. People tried to target metabolic pathways in cancer cells by using inhibitors of glycolysis and other pathways. So when I first heard this in a lecture in a meeting of the UK Rheumatology Society way back in 2018, the first thing that came to my mind was, how can you block something that is so fundamental to life? So when I asked the speaker this question, they said that, you see these pathways, there are so many different pathways which cover for each and every process. So there is a lot of redundancy. Hence, if you block certain pathways which are overexpressed in certain cells, it is likely that those cells itself would go down in number or would die off, but the other cells would not be affected. So that's the entire concept of immunometabolism, that immune cells have perturbations of these metabolic pathways, which in turn affect their function and help in the preponderance of one immune cell subset over the other, say a balance between pro and anti-inflammatory immune cells for that matter. So metabolic pathways are basically of three types. If broadly speaking, glucose metabolism, fatty acid metabolism, and amino acid metabolism. So glucose metabolism, there is conversion of glucose 6-phosphate to pyruvate, which in conditions where there is lack of oxygen can go for anaerobic metabolism to form lactate, or in the presence of good amount of oxygen, it can enter the mitochondria forming acetyl coenzyme A, go into the tricarboxylic acid cycle, and then into the electron transport chain, etc. Similarly, for fatty acid oxidation, it generally goes into the acetyl coenzyme A and into the mitochondrial metabolism. And for amino acid, glutamine via glutaminolysis is converted to alpha ketoglutarate, and this again enters the tricarboxylic acid cycle. This final oxidative phosphorylation that occurs in the mitochondria leads on to transport of electrons to the electron transport chain. And also there is this pentose phosphate pathway which deviates from the glucose 6-phosphate to form nucleotides for the synthesis of nucleotides. So as we're understanding in immunology, there are these different cellular populations. Let's say, for example, T lymphocytes are one of the common subsets that are involved in a variety of immune processes. So there are T helper 1 cells, T helper 2 cells, T helper 17 cells, and T regulatory cells. Each of these cell populations shows a different preponderance of these different metabolic pathways. For example, T regulatory cells show more of mitochondrial metabolic pathways. And particularly the balance between T helper 1 and T helper 17 cells sometimes drives different disease processes, because T helper 1 cells essentially help to target intracellular organisms. T helper 17 cells attract neutrophils and bring in inflammation at sites of infection. So T helper 1 cells are generally having a preponderance of formation of pyruvate to lactate, and T helper 17 cells have more of glutaminolysis and fatty acid oxidation. So this is just an example of how these metabolic pathways differ in different cell populations and can lead on to different balance between different cell populations.
Thank you, Dr. Misra. So if I understand correctly, it would also matter which particular pathway is used, like initially how the immune response starts. So is it a different pathway used at that time versus something in the adaptive immune system? Maybe if you can just briefly explain the immune response to SARS-CoV-2 for non-immunologists. So would that also, and do the immune cells have an ability to switch from one to the other? I'm sure they do. And of course, the
Yes.
important question is, how can we utilize that for treatment? So I understand in the other world of your autoimmune diseases, those are some of the drug targets. So first and foremost, when the first line of immune defense, will that matter of which particular pathway is used versus later on in the adaptive immune system? So is there a difference between the specificity and the efficiency and the way in which any of these things happen?
Yeah, Dr. Kar, if I will take up this.
Yes, please, Dr. Agarwal.
So what you are asking is very fundamental, but somehow we still don't know which is the predominant metabolic pathway and which is the predominant cellular immune responses which are there. But what we do know is that patients who have got a very mild to moderate disease of or infection of this SARS-CoV-2, they have got not significant lymphopenia.
Yes.
The number of circulating CD8 T cells which are able to kill the virus are sufficient. Similarly, their peripheral blood does not have a reduction in the B cell number or CD4, CD8 T cells, which express CD38 and HLA-DR. Basically, these are activated T cells. And similarly, it has also been reported that patients who have got mild to moderate disease, their residential T cells in the lung, they are very much efficient and they are very much active and they can kill the virus very efficiently. Also, the number of macrophages and the dendritic cells in these subjects, they are very much near normal or activated and sufficient to kill the virus. However, when you talk of patients who have got severe disease, and I'll come to that because this is beyond metabolism or metabolic pathways, how we can explain the mild to moderate or severe disease. There, the number of immune defense cells are very much reduced, like there is reduction in dendritic cells, T cells, NK cells, even monocytes and macrophages in the circulation. So, and whatever B cells are there, they are not producing a lot of neutralizing antibodies. The plasma cell number is also reduced. And the phenotype of natural killer cells, which are there in the peripheral blood of severe COVID, they are very much not active or exhausted kind of a phenotype. And the CD4 cytotoxic T cells, which are there in the circulation, they also are not able to kill the virus very efficiently, and especially those which are infiltrating into the lung also. So clearly, there is a difference between the immune response to mild to moderate or a severe COVID infection. Now, why this is happening? Probably this lies in the fact that subjects who are having severe disease, they are having either a heavy dose of the virus, or their natural immune responses were very poor. That is why the virus is able to multiply. Like we know that elderly patients have got immune senescence, so their immune responses are not that great as compared to the younger people. So the virus persists. And also we do know, when the virus is entering the airway epithelial cells, and if the type 1 interferon responses are poor, or virus has mechanism to evade type 1 interferon responses by blocking the rig-like receptors which are present in the cytoplasm, which recognize RNA of the virus and mount an immune response, they these viruses, they survive, proliferate in the cell, and while proliferating, they cause damage to the cell. Now, these damaged cells will release a lot of damage associated molecular patterns called as DAMPs. And DAMPs are very intensely pro-inflammatory signal for the body. So these will activate a lot of metabolic changes in the immune cells which are already there. Remember, still we are learning a lot about the virus. We have not understood this virus completely, because this is very much different as compared to the other beta coronaviruses, especially the original coronavirus, or even Middle East respiratory virus, or our garden variety H1N1 or other influenza viruses. So we are still learning a lot about this virus. So probably this is the reason why this virus has got, probably this is peculiar to the virus, that it is causing, because of some host factors, it is causing mild to moderate disease or asymptomatic disease in some people, and in a very small proportion of people it is causing severe disease. Yes, people have reported that patients who do have a severe disease will have a different metabolic programming of their immune cells. As Dr. Durga has already elicited, all the mechanisms which are there, predominantly it is the glucose metabolism which is affected. And that too, the glycolytic, or what we call as aerobic glycolytic pathway, is very important. All the immune cells, whether they are T cells, B cells, or monocytes, or especially of M1 phenotype, they all undergo a process of aerobic glycolysis, also called as a Warburg phenomena, which was almost known about 100 years ago. So, so this is how things are actually. And during this, what is this aerobic glycolysis? Because we all know that one molecule of glucose can generate 38 molecules of ATP.
Yes.
Two of these molecules will come from the glycolytic pathway, whereas 36 molecules will come from the Krebs cycle or the tricarboxylic acid cycle. So we all know that for an efficient system, tricarboxylic acid cycle is very important. But in conditions where this inflammatory milieu is happening, the immune cell has to adapt to the worst case scenario. And the worst case scenario will be, there will be hypoxia, there will be less of nutrition. So the mechanism has evolved, probably it is evolutionary only, that immune cells are able to survive in these harsh conditions, and still they are able to mount an inflammatory response to counter the threat.
Yes.
That is why they have adopted this policy of going to aerobic glycolysis. And during this process of aerobic glycolysis, many things are happening. Cell is able to withstand the hypoxia part, and it induces generation of hypoxia inducible factor alpha, which will be speaking about in a couple of slides ago after this. And also there will be a lot of changes which are happening, which will push the cell towards an inflammatory phenotype, where a lot of reactive oxygen species will be generated. There will be stimulation of mTOR pathway. Now, mTOR is basically a mammalian. Uh,
Mechanism.
yeah.
Yes.
So basically, mTOR senses the metabolism of the cell, and if cellular metabolism is going haywire, it will be activated. will try to protect the cell, and that is why the mTOR pathway is activated. Now there are two complexes of mTOR pathway, mTOR1 or mTOR2, and depending on the cell, immune cell, this will be activated. mTOR1 is basically for pro-inflammatory signaling.
Mhm.
It also leads to activation of a lot of pro-inflammatory cytokines and these IL-6, IL-8, IL-12, IL-13, IL-23, which will be, there is a list of cytokines, no need to remember for anyone, where the inflammatory cascade will be, and a lot of chemokines also, so that the inflammatory cells come together to try to mount an effective immune response against the threat or the pathogen. And here we are talking of coronavirus 2. So this is how probably the metabolic programming is happening. Now, when the aerobic glycolytic pathways are activated, the other pathways, they take a back seat. The tricarboxylic acid cycle becomes less prominent. And but yes, there is something which is happening in the amino acid metabolic pathway, which favors again a pro-inflammatory milieu and a pro-inflammatory response mediated by the immune cell. I think it's a fairly lengthy response to your very small question.
No, Dr. Agarwal, thank you so much, because for non-immunologists, that is a lot of information to digest, first and foremost. And the other one important factor I wanted to bring up is, we know that the immune system is highly sophisticated and a big energy user. So we talk a lot about, the brain using up about 20% of the body's energy supplies. And if I am not wrong, I think the immune system, to mount an acute immune response, uses about 20 to 30% of the body's energy sources. Am I right in that? So
Yes,
it's highly energy dependent, number one. And another thing that I just wanted to briefly mention, because this webinar is not about vitamin D, but we've come across a lot of studies where vitamin D deficiency has been a factor in, higher susceptibility to the, to poor outcome in the COVID-19 pandemic. And you talked about the, the dendritic cell. So I think vitamin D is an important modulator of the dendritic cell. Of course, that's not the only factor. So is that one area where the immune response would be different in someone who's vitamin D deficient versus who is replete with vitamin D?
definitely, because a lot of immune cells, including lymphocytes,
Mm-hmm.
they have vitamin D receptors on their surface. And general response of vitamin D is towards uh, decreasing the inflammation or anti-inflammatory response. Like it is like glucocorticoid. Its structure actually is very much like glucocorticoids
Mm-hmm.
itself. So it has got a immunosuppressive effect. So if there is a deficiency of such an vitamin, obviously, and the milieu is all pro-inflammatory, the chances of having and in the this infection of this SARS coronavirus 2, most of the things are going downhill because of our own immune exaggerated inappropriate response rather than because of the virus itself.
Thank you. And that is extremely important, because again, there are two areas in this, which is sometimes, people, not people who know what is going on. A lot of lay people, they think that, okay, vitamin D is supposed to be good, so I should take, super high doses every day. Now, that is also not what is recommended. And another big difference is, because people talk a lot about vitamin D, there is a difference in using vitamin D to build up immune resilience versus using vitamin D as a treatment of COVID-19. Now, we are not going into that. I think one paper came out from PGI Chandigarh where they used very high dose of vitamin D as treatment, not in those. And in India, everyone's vitamin D deficient unless they are taking a supplement. So that was,
Yeah,
anyway, my point was not to digress into vitamin D, but vitamin D is so common and so, relevant. So that's what. Another thing, important thing, Dr. Agarwal, you mentioned about the natural killer cells, and I think, Dr. Latika, we had done another video on estrogen and how it gives you immune resilience, and we talked about how sleep deprivation reduces the natural killer cell activity, and that is why I talk about, people talk about building immune resilience. One of the first things you can do is sleep well. And Dr. Latika, would you like to add anything to what Dr. Agarwal has said already? And Dr. Misra, same to you. Would you like to add anything, or will I go ahead?
I totally agree that hormones do have a role to play here as well. And I think we discussed that at length even previously.
Yes.
And we'll be covering it. Yeah, this is a slide. So clearly there is a difference in mortality between men and women. And these trends emerged early on and they have been confirmed in subsequent reports. And like we were talking about vitamin D, estrogen is no exception. So lymphocytes do bear receptors to estrogen, that is estrogen receptor alpha and beta both are present. And people have shown that in premenopausal women, mortality trends are much lower, and in postmenopausal, it's higher. And there's an inverse correlation of cytokines with the estrogen levels. And estrogen can also affect the T helper responses and NK cell responses, which we talked about earlier. While with testosterone, the relationship is more complicated, like it is in general thought to be more immunosuppressive. But men who have very low testosterone levels also have higher susceptibility to COVID.
Yes.
So, but another interesting aspect is the viral entry into the cell. So this has been studied and in the setting of pregnancy. So people were initially worried what would it be like pregnant women and COVID would be a major problem, but the estrogen levels are high and these women do extremely well. And similarly, there are also in vitro studies where it is shown that when you have very high estrogen levels, then the AC2 receptor levels come down. So probably the entry of virus itself is abrogated, and that may be one of the mechanisms of protection. So at that time, if you remember, we discussed some of the trials which were ongoing with estrogen patches and some of the androgen antagonists. So I looked up again, and one of those is published now. I think not those, but one of the retrospective data sets where they have looked at postmenopausal women who are on estrogen supplements, and the risk of severe COVID is 50% lower. Yeah. So the odds ratio is around 0.29 for these women who are on estrogen supplements. So clearly there is a role, although this is not everything, there's a strong genetic component to it. But this is one thing to be mindful of and maybe possibly exploited. Time will give the answer to that question.
Thank you, Dr. Gupta. And in the interest of time, we are not going to go into the hormones because we've done a separate video. And there is, when you talk about those women who are on supplemental estrogens, one big area is, are they on oral estrogens versus transdermal? Because oral estrogens are highly inflammatory. Transdermal are not inflammatory because we have to think of the heart as well. So this is one differentiation that a lot of papers don't do, a lot of doctors are not aware. If you ask, regular OBGYs, even all of them are not familiar with this. Then the other component comes with the progestogen component. Is it a medroxyprogesterone acetate versus an oral micronized progesterone? The two are completely different. The medroxyprogesterone acetate is also a mineralocorticoid receptor agonist. So all the bad effects of hypertension and all. Anyway, we're not going into that. So this is just a summary of the ones that we have any data for. Now, I just wanted to bring up anemia because iron deficiency anemia is so common in India, and all of you have been at the COVID front line. So did you find that all those people with iron, and even in men actually, iron deficiency anemia in India is not as uncommon. Did you find that they did much worse?
So I wouldn't say that we've looked at data as such, but this was one of the early hypotheses which came up, particularly with relevance to India, because where worldwide data suggested that women do better, in India it was the other way around. Sort of thing as well. Yeah, but then so one of the theories doing the rounds was that it is anemia and probably poor accessibility to healthcare and other social issues. So anemia was really postulated as one of the factors, but I wouldn't know if that is indeed, or if someone has systematically looked at it, but definitely it seems plausible because it will affect directly the oxygen carrying capacity of the RBCs and hypoxic state and overall poor metabolism.
Thank you. Dr. Misra, would you like to add anything to this discussion now, or shall we go ahead?
I wouldn't have any data to look at iron deficiency, but offhand, most of the admitted patients with COVID pneumonia, they have good hemoglobin levels. And of
Okay.
course, it's difficult to assess iron deficiency because of hyperferritinemia, which
Yes.
occurs due to the inflammatory
Okay.
response. So
Yes, we
it's
won't have time to go
really do
into the
not.
hyperferritinemia today. So basically another thing I wanted to highlight for, see what happens in India is whenever people find, anemia based on a diagnosis of just looking at a CBC, not looking at ferritin, not looking at serum iron, not looking at TIBC, none of the iron studies. One important factor is there is an anemia. I mean, I'm not talking to you all, it's for the others, which occurs because of a disease. It could be an acute disease, it could be a chronic disease. And an anemia of chronic disease, all of you you're dealing with it all the time. That is something quite different from an iron deficiency anemia because of insufficient intake of iron, or iron is very important in the immune system as well. So my point is, a lot of doctors may see anemia and without deciding whether it is an iron deficiency anemia or is it an anemia of disease or chronic disease or acute disease, giving them iron supplements may not always be the best thing. Am I right in saying that?
Absolutely.
Okay, so I'm going to the next one. The most important question we had is because now we know that in India, diabetes is so common. It is already the diabetes capital of the world. And I think about 75 million people with diabetes by 2025. And just in context, the entire population of Canada is about 37 million. So going by that, now this was a paper which I think this was from Kuwait, where they found that the fasting blood glucose levels, and these were people who didn't have diabetes before, they were not diagnosed as diabetes before admission. What they found is fasting hypoglycemia, and this was not like, super high. This was about 140 milligram per deciliter. And they found that they did much worse. That was one thing. The other thing is even minor changes. So this was, I think they reported in millimoles per liter. So this was 6.9 to 7. Even that tiny change, tiny increase made a big difference to the prognosis. That was one thing. So I'm sure you've come across similar, you've had similar experience as well. Then the other thing that that came up was, which we are going to talk about later, is number one, this would definitely decide the management in the ICU. And I'm not an intensivist, but if I had an option, this is again something I'm just throwing out an idea. Would it be a great idea to have a continuous blood glucose monitor on all these patients in the ICU? What do you all say to that?
Yeah, actually, we had a closer look at the data of this study that
Hmm.
you have showing here. This is a study from Kuwait, if I remember correctly,
Yes. Yes.
and some data on some 400 odd subjects were there. And they initially modeled this for a linear
Yes.
model, whether they could see if that was negative. Then they went on to look at a non-linear modeling, and then they could detect that yes, from 6.9 onwards, the even within that range also, those who had a lesser level of blood glucose, they did well as compared to the ones which were having the higher blood sugars level. uh, Difficult to say actually, because uh, we had also seen a lot of patients in last 9 months at our place, but we didn't come across such a minor rise in blood sugars and the adverse outcome.
Okay.
So, but we give credit to the authors of this paper
Yes.
who think out of the box, and they proposed that a very strict blood glucose monitoring should be done. Now,
Mhm.
the problem with blood glucose monitoring in such a scenario is, like, there are other reports where if you use insulin to
Yes, I'm coming to that,
glucose
Dr.
level, yeah,
Two
then the
slides
things,
down the line, that is what we are going to come up with. So just to give us this thing of what we are going to talk about later. So There were other studies which showed that those patients who were given insulin in the ICU, they did much worse. And we'll talk about insulin and immune system modulation later on. And the only thing I was talking to an intensivist friend of mine, I said, I am so glad I am not making those decisions, whether should I give insulin, should I give something else? Because in the ICU, insulin is one of the drugs of choice for managing hyperglycemia. Anyway, so this was again an in vitro study. So I will, who's going to talk about this one between the three of you?
Yeah, I'll take it up.
Okay.
So, so this sort of a problem that whether patient has got hyperglycemia and how to manage these patients and whether it is going to affect the outcome at the end of the day has been very much questioned word over in many studies. And people were not able to link this hyperglycemia with the severity of the infection. But what it has been realized now that hyperglycemia actually causes increase in inflammation. It also helps in the survival of the virus within the host cells, and it causes expression of glucose transporters, whereby the glucose is taken up into the cell, which actually helps the especially the immune cells in undergoing an aerobic glycolytic pathway, where they lot of hypoxia inducible factors and the other genes which play a role in inflammation. And actually, inflammation is causing the adverse outcome in most of the subjects. And here we are talking of monocytes, actually, which are believed to be the at the boundary of the innate and the adaptive immune system. And these play a very important role because these are the first amongst the first line of defense, as well as they give or transfer the immune response to adaptive immune system later on. So here it was clearly demonstrated that once the virus is actually getting into the uh, monocytes or macrophages, it is somehow causing the blocking of the type 1 interferon responses. And now this type 1 interferon response cascade is a bit complicated, but in simply, once the virus enters, it is recognized by the cytosolic proteins, which are RIGS or MDF5, and these causes downstream signaling, whereby interferon release factors are activated, which causes release of type 1 interferon. And type 1 interferons are basically interferon alpha and beta. Now, these type 1 interferons, they in an autocrine fashion stimulate the very cell from which it is being liberated or released through interferon alpha receptor 1. And once it activates these interferon through activation of cyclic GMP, AMP synthase, and the stimulators of gamma interferon or the genes, which are called as stings, which causes release of type 3 interferons. Amongst the type 3 interferons is basically interferon lambda, which has got a very potent antiviral effect. Now, if there is any blockade in RIGS or downstream signaling or blockade of IRF or blockade of interferon alpha receptors, this cascade of interferon response is blocked and it helps the virus to survive. And once the virus is surviving, it is causing all kind of metabolic reprogramming of this very cell, and which is trying to survive the virus onslaught. For this, during this process, what is happening is the uh, cycle is going into the glycolytic aerobic cycle. There is a lot of hypoxia inducible factor expression, and there is expression of ACE receptors also on the surface, which actually again helps the virus to survive. And people have shown that once this is happening, as the virus load is increasing, there is increased expression of the genes which are playing a role in the glycolytic pathway. This is a very elegant paper which has demonstrated this. And also a lot of pro-inflammatory cytokines, as well as ACE2 receptors also, which are getting expressed. And again, this whole cascade is fulfilling the survival strategy of the virus and harming the host. So this is clearly established that once you have a viral infection and somehow weak type 1 interferon response, the virus will survive. There will be increased expression of glycolytic pathway genes, increased expression of ACE2, increased expression of pro-inflammatory cytokines, which are going to be detrimental to the host.
Thank you, Dr. Agarwal. This was a very elegant paper, and this is a paper done on the COVID-19, the SARS CoV-2 virus. So just for those people who are not into research, usually what happens is people come up with the hypothesis like, okay, glucose is something that impacts immune system. Then what they do mostly is they do what is called an in vitro study. So in vitro study is a study in a lab this is what this study was. And then the next thing comes the clinical application on, so here from a study like this, the application would be, does reducing blood sugar levels in patients with the COVID-19 infection, does it impact their prognosis? Yes, it does. So the other reason of doing these kind of in vitro studies in the lab is to find out where exactly what is happening. Is it a problem of the ACE2 receptor, challenge? Is it an, IL, one of the interleukins or any of that? And for those of you who are not immunologists and in the audience, or you're going to listen to all of this later, a lot of this information you may find overwhelming. That is because you're not familiar with the immune system. Neither am I. I had to go back and study. So people of my vintage, we didn't study much about the immune system in medical college. And as an OBGY, we actually probably studied almost nothing. So my point is, don't become overwhelmed. All that information is there. But then it is very important because it's not a simple thing of, oh, this is just this and this is just that. Sometimes we have this reductionist mentality of, oh, just take high vitamin D and everything will be fine. No, it's not that. So the other thing is, again, the same from the same studies where the ACE2 and the lung macrophage, and I think Dr. Agarwal, you already covered that. And Dr. Misra and Dr. Gupta, would you like to add anything more to this?
No, I think we've covered it all.
Okay. Dr. Misra, anything would you like to add?
No, nothing really. I think
Okay,
Professor
thank
Agarwal
you.
has covered this.
So the other thing is the mitochondria. Now, why am I bringing up the mitochondria in the immune response? For most people, that the mitochondria is a very important organelle in the cell for generating energy, generating ATP. And why I bring it up is, people with long COVID. So long COVID is people who have recovered from, the coronavirus infection. And some of them have recovered from a rather mild version of the disease. They were not in the ICU. They didn't even need, some of them were self-isolating at home. But what they found is, and again, it depended. So by definition, long COVID is more than 3 weeks after the infection. They felt that they had a lot of symptoms. One of the commonest symptoms they talked about was fatigue. So that is the reason I'm bringing up the mitochondria, because in my world of functional medicine, we look at mitochondrial bioenergetics as one of the factors deciding on fatigue. And all of you as immunologists, you are very familiar with fatigue. So just briefly for a non-immunologist, how would you explain how the mitochondria is associated with this response? And more importantly, what can it do long-term, like for someone with long COVID?
Yeah, a very sort of a burning problem right now for which we don't have any kind of an answer. But just to explain to the audience, mitochondria we all know is the source of the energy, and we call it a sort of bioplast. This was named as bioplast by Dida, and later on called as mitochondria. But it does play a very important role, and it is a sort of a part of innate immune response also, especially for the viruses. When I say this viruses, I'll explain how. So once the virus is entering, and as I mentioned
Hello, I think we lost it.
MCA5, the
Dr.
next
Agarwal, I'm sorry to interrupt you. We didn't hear the last few words.
okay, yeah, I'm my apology for the weak
No worries.
connection.
Yeah, don't worry. That's okay.
So once the virus enters the cell, it is recognized by the cytosolic proteins like RIGs, which are called as RIG-like structures, or by MDA5. Once it is done, then it activates mitochondrial antiviral sensing or signaling proteins which are present on the outer mitochondrial membrane, which is also called as MAVS. Now, this mitochondrial antiviral signaling protein actually brings downstream cascade, activates interferon response factors, which actually bring out the transcription of various type 1 interferon genes, whereby the interferons are released by these cells. Now, if there is any sort of uncoupling of this MAVS, or if there is any blockade of this MAVS by the virus proteins, because it has been known in original SARS corona infection that there are certain nucleocapsid proteins which can block this signaling and which will not allow the cell to cause type 1 interferon responses to happen. So this can happen, and that is why the cell in the virus will survive in this particular cell and will cause on lot of inflammation, which will ultimately lead on to generation of lot of reactive mitochondrial oxygen species or reactive nitrogen species, which will or will cause more of a inflammation. So this is how the virus is actually having an impact on the mitochondria. Besides this, when the aerobic glycolytic pathway is active, so in the aerobic glycolytic pathway, the last metabolite formed is pyruvate. Now, this pyruvate can become can go into a acetyl coenzyme A pathway, can be actually go into a lactate pathway, as Durga has told in the initial part, and can enter the tricarboxylic acid cycle. And there are again certain intermediates of these which will end up in succinates. Again, these succinate can also help in the tricarboxylic acid pathway and the other pathways also. So this is how mitochondria has a very important role to play in the viral infections. Now, once you say that viral infection has happened and the mitochondria are activated, so what happens? The mitochondria can has a property of undergoing a fusion or a fission.
Okay.
Once the hypoxic conditions are there, the mitochondria can actually fuse together to form long organelles. And this is generally happening when you have a severe COVID-19 infection of a cell. When there is a lot of oxygen or adequate amount of oxygen is available, then they undergo fission and they can form small mitochondrial complexes all over the cell. So during this process of long organelle formation, it is trying to mount an adequate energy supply to the cell as well as adequate immune response so that the cell will survive the onslaught of the virus. But it may not happen altogether. So at the end, the mitochondria may be damaged. And once the mitochondria is damaged, the mitochondrial DNA, which is released into the cytoplasmic cytosolic compartment, will lead to further activation of the sensors which are present, which recognizes DNA, think it to be a danger signal, and again will lead the cell towards a inflammatory response, which will require a lot of energy and the nutrition also. So the things will be going on like this forever. So probably to explain why the fatigue is seen more in long COVID haulers, probably this is the response that their mitochondria number is decreasing, and whatever the mitochondria are available, they are not sufficient. Either they are using sufficient amount of energy and nutrition for fulfilling the cellular metabolism to survive, or they are unable to produce a lot of energy for the cell to mount an effective function of the cell, and which it is causing a lot of fatigue to the person.
Thank you, Dr. Agarwal. I'm going to rush a little bit through this because I'm just being a little cognizant of the time. So this same paper that we've been talking about, what they also did was they also looked at adding things like coenzyme Q10 and N-acetylcysteine. And why I'm interested is because these two are things that we use to bring out, to treat people with fatigue. So what does coenzyme Q10 do? And then again, this was an in vitro. This is a study in a lab. So can we use it in people? Yes, because coenzyme Q10 is a supplement. So when we talk about supplement, it means the body is naturally supposed to have coenzyme Q10 in the body, versus using a herb is something that has to be given extraneously. So there is a big difference between supplements and herbs, and sometimes people don't get. So a curcumin is a herb, coenzyme Q10 is a supplement. Now, the other interesting thing is, with age, coenzyme Q10 levels go down. So definitely that would impact mitochondrial bioenergetics. Then there are some medications which deplete coenzyme Q10, statins and beta blockers being two of the major ones. So, and given that age, added, so when we talk about medications, now having any comorbidities would definitely impact prognosis in COVID-19. And we know that comorbidities like hypertension and heart disease. So it is also possible that these people have, and I don't think anyone has looked at the coenzyme Q10 levels in blood levels in these people yet. Maybe some studies will come up. So what they did is they looked at, this was again an in vitro study where they added these and they found some beneficial effects. Now, where do coenzyme Q10 and N-acetylcysteine fit in? Now, NAC is also an approved supplement available, and I think in SLE and some of these other autoimmune conditions, NAC has quite a lot of data. Mental health, there's a lot of data on the use of NAC. So this is again the mitochondrial in the inner membrane that Dr. Agarwal mentioned a little while ago, and the complex 1 and all the 4 complexes. So here, this Q is the coenzyme Q10. This is where it works. The other thing I wanted to talk about is metformin is supposed to be an anti-aging drug and a lot of things. And of course, metformin is used in diabetes. And metformin works on complex 1. Is that correct?
Yes.
And like I was talking about, statins causing depletion of CoQ10. So this is again one area where, it's not just, putting someone on a medication. It also has other areas that we need to look at. So in my world, if someone is on a statin medication, and again, I'm not going into who needs to be on it, doesn't need to be on it. Do women need to be on a statin? We don't have the data yet. But the thing is, important thing to bear in mind is we do need to supplement with a CoQ10. And again, we use a lot of CoQ10 for dealing with fatigue. This is one. The next thing is, where does N-acetylcysteine come into this? So this is the methionine homocysteine cycle. And again, methionine is one of the amino acids. So homocysteine again can be easily measured in blood. And high homocysteine is associated with higher rates of cardiovascular disease, mental health challenges, dementia, everything. So in this cycle, you can see one is, of course, in green, there are a whole bunch of B vitamins, which are very, very important. And here, homocysteine has to form cysteine, from which glutathione is made. Now, glutathione, as all of you know, is a very, very powerful antioxidant. So what happens is, if homocysteine is high, it is possible that it's not making adequate cysteine and glutathione levels would be low. And one of the important things is, folate is very important. And I'm not going into that entire, cycle. But one thing I want to stress upon is the B12 deficiency, which is very common in India because a lot of people are vegetarians. And drugs like, metformin also depletes B12. But the fact is, most of the time, we are only measuring B12. We are not measuring folate and B6 and all the other B vitamins. So when we find a B12 deficiency, very often it is not just a B12 deficiency. However, in vegetarian people, because they eat more of folates, a lot of dark green leafy vegetables, usually the folate levels are not low. And again, folate is not folic acid. I'm not going into that. My point is, usually if there is a B12 deficiency, often it is not just a B12. It is also accompanied with all the other B complex. But the point I wanted to make here is, when they looked at N-acetylcysteine, this is where, cysteine is, provided by the NAC. Because the other challenge with glutathione is oral glutathione, unless it's a special form, doesn't always work as well. So most often we talk about maybe they need an IV glutathione or maybe even glutathione transdermally. So these were just two things that I just wanted to highlight. And next we are going ahead with what is the role of insulin in immunity. So this is the study that we talked about before, that people who were given insulin, they did much worse. So basically what happens is, now the diagnosis of diabetes is basically just dependent on whether the person has hypoglycemia. Now, hypoglycemia can be achieved in many different ways. Is it from insulin not working well? Is it insulin clearance that is not well? And we know that in people of South Asian ancestry, they lose beta cells at a much lower blood sugar levels. However, here what we are talking about is how does insulin affect the immune response. So who is going to take up on that?
Yeah, definitely insulin is always on a clinician's mind whenever there's uncontrolled diabetes, because there's potential to reverse some of the damage done by hyperglycemic state. But the authors of this previous study, the one on the previous slide, it was really interesting that this Chinese group, they found in 600 patients, it was a retrospective data set where they found that the group that received insulin had higher mortality, which is goes against what one would expect. And at first, if you were to look at it, you would consider it to be a selection bias, but the authors have gone to great lengths to check and validate their observations. So they have addressed this using a Cox regression and then later done propensity score matching. They've also done subgroup analysis, various timing groups, and despite that, there was clearly an increased mortality in the insulin subgroup. And I would say that in light of all these robust statistics, the data should be taken as a serious warning, even if causality is not proven yet. So one interesting finding in this paper was that those who had higher, those who were on insulin also had more episodes of hypoglycemia. So it almost makes us think if that is the cycle that reboots the entire metabolic process and that is causing damage. But then they also analyzed the group that did not have hypoglycemia.
Yeah.
And the other interesting thing, we did talk about metformin in the previous slide. So the group which was on metformin had lower mortality and they had better survival. But then later they also analyzed those who were not exposed to metformin. And again, insulin clearly was one of the, seemed to be associated with higher mortality. Again, the group which was only on insulin versus insulin plus OHAs. So even though the second group had higher glucose levels, they had better outcomes. So clearly there was an association which could not be explained. But there are papers which suggest that insulin can directly activate macrophages and increase cytokine production through the NLRP3. So that is what, so here we don't know what the triggering factor is. Is it hypoglycemia, or we go to treat it with insulin? So insulin is a causative factor. It's really difficult when the inciting event itself is nebulous and can be a very challenging situation to deal with in the ICU. That's something which definitely needs to be looked at carefully.
Yes. And again, none of us are intensivists here today. So I was asking an intensivist friend of mine, I said, like, how are you all dealing with this? Because on one hand, you find that giving insulin, which is usually the standard of treatment for any patient in the ICU to manage hyperglycemia, if this is going to cause this problem. Another important thing that she mentioned was the use of glucocorticoids and whether giving higher dose steroids made a difference, because on one hand it was used as a treatment and it helped a lot of patients, but we know that glucocorticoids do increase blood sugar levels. That was one factor. And another factor that I was thinking is it will also impact how the IV fluids are decided. And again, I don't think we probably don't have any intensivists in the audience today, because the IV fluid management in the ICU would also matter, because on one hand, does lactate help in the SARS CoV-2 infection? Because lactate is also very important in the bioenergetics, right?
Yes, definitely. But this study exemplifies that we need to see how we are managing glucose. It's not just managing it, because even metformin has its own problems. It is associated with lactic acidosis. So especially in the critical care setting, even though one of the studies has shown some decrease in mortality only in women. But then those patients typically did not have all the end organ damage and associated comorbidities, which can be difficult to deal with if diabetics have those.
So yes, as you rightly pointed out, Dr. Gupta, that how normoglycemia is achieved. And we know that there is a lot of data coming from the diabetes world, that blood sugar is a biomarker, but the thing is, we know that hyperinsulinemia, the endogenous insulin that is produced, is a very highly inflammatory molecule. But exogenously given insulin is also possibly very highly inflammatory. Now, do the immune cells have an insulin receptor? I think they do, right? Dr. Agarwal, Dr. Misra, would you like to add something
Yeah,
about
they
the
do.
insulin response, the insulin in relation to immune cells?
Yes, insulin does have receptors on the immune cells, and it has been known, in fact, not only in the this SARS CoV-2 infection, even in other ARDS also, insulin does actually promote more of inflammation.
Mm-hmm.
So there is already inflammation happening and damage happening because of that. So it actually, rather than helping the patient, it is harming the patient.
Yes.
Insulin promotes glycolysis, and with this glycolytic switch, there is a increased shunting of glucose down the pyruvate pathway. There's also increased ROS generation. So as we talked about initially, whenever there's more oxidative stress, then it will trigger inflammatory cytokine production, both directly via the interferon pathway and also via the inflammasome.
Okay, thank you. So this is a question that I wanted to ask you. Now, I know all of you have done numerous studies where you looked at the proteomics, metabolomics. So I would ask you all to explain what is proteomics, what is metabolomics? And the second question is, can we use lipid abnormalities as surrogate markers? So we'll come to that. So proteomics, metabolomics, for people who have no idea what this is, they hear a lot of things about big data and omics. From the world of omics, a lot of treatment modalities are going to come up. So please will you explain for non-immunologists, people who don't know what this is, what is proteomics, what is metabolomics?
Yes. So
Okay. Yeah, Latika, go ahead.
So metabolomics is basically, it is the final common pathway. It is a study of the metabolome. So all the metabolites together, which are present in the environment and which are the final common product of all the pathways that are operating in an individual. So basically hypothesis-free approach where we just pool in patient samples and we look at the entire metabolome, the spectrum that lies within, and then we analyze that all together. So it's a study of small metabolites, which are typically less than 1 kilodalton in size. And being the final common pathway, it is also what has actually happened rather than what we project that, okay, genetics is what, genomics, what may have happened, or transcriptomics, this may have happened, but metabolomics is considered to be the final common pathway. Same with proteomics, it just lies above that. So clearly, these have been studied in many different diseases, and COVID is also something people want to look at it. But the problem when you analyze this kind of data is it can be highly heterogeneous. So there are various different techniques. It can be using LCMS, or it can be done using NMR. So people are looking at open data sets and collating all the data together, making uniform methods, looking at the techniques, and also looking at the spectra rather than the final analysis. So it is very important because it's an extremely sensitive technique. And although we can pick needles out of the haystack, but we need to know how important those are. So methods need to be valid, and this is being looked at by people through spectral analysis in open access data sets.
Dr. Agarwal and Dr. Misra, please will you add to that?
Yeah, actually, what Latika has told is correct. Actually, there are a lot of omics to modern day research and science, and absolutely right in the direction also, because probably the sum total of understanding of all of these will give some answer. And we can add even transcriptomics also, if we can take some cell and then look at what all kind of mRNAs are actually active, which might be telling us the story how the cellular metabolism is actually happening right now. And even newer technique like single cell sequencing has also been available now to most of the researchers. So you get a lot of insight, into a particular type of cell and what kind of a what kind of a metabolic alterations are happening. And we can know from the data set of thousands of up and down regulation of various genes and their splicing variants, what kind of a metabolism is happening, which will may suggest like this may be happening because of this or that, and how to regulate it or how to control it later on. For proteomics, similarly, we can have an approach where we can look at what all kind of peptides or the amino acids are or their metabolites are actually being altered when we are looking at the cellular metabolic pathway. Just for an example, we know that when people looked at certain metabolites in the plasma of patients of severe COVID-19, they could detect a lot of metabolites of tryptophan, and they could find a lot of kynurenine or kynurenate and the other metabolites of tryptophan. And that is how they could relate that, okay, this is an essential amino acid which is depleted in patient with COVID-19. And this might be having some role to play. And that is how they could come up that probably when you are talking of nutritional or translational potential of this kind of an information, then we know that we can supplement with essential amino acids to these patients during this process of severe stress or immune metabolic changes which is happening in their disease course. So this is all, otherwise it's a very sort of a complex technology, and rather than performing the test, it is more of a more complex when you analyze the data actually. So like what Latika said, it's like a needle in the haystack. You have to make head or tail out of it once you have a plethora of information with you. And this is how things are happening. And a lot of information has come up in last around, say around 15, 20 years with the use of this omics approach. And I think, however, for as far as COVID is concerned, not many omics data are available
Yes.
because problem is you can't procure data as freely as you can do for the other diseases. Number one, you need a BSL-3 or above certified labs where only the samples can be processed. And so these type of labs are not very many in the world, and they might not be doing this kind of a research actually.
Thanks,
But
Dr. So, Dr. Misra, would you like to add something to that before I talk about what is essential amino acids and the other hypotheses that I would love to talk about? So, Dr. Misra, anything you want to add?
Yes, metabolomics is emerging as a adjunctive tool in understanding a lot of immune-mediated rheumatic diseases. And the only issue being that whether these are associated changes or whether these are causative changes, that's what is difficult to find out in a cross-sectional study. I think most of the other points have already been discussed.
Thank you. So for just a recap of what, so proteomics, metabolomics, and like I said about can we use them as biomarkers? Now proteomics, metabolomics, these are not tests that are available easily in just someone goes to a lab and I want to do a metabolomic study on myself, you can't do that. So these are not available as clinical tools, they're more research tools right now. And the other important area, which I think this is what, going by my brief this thing of the literature, in the future, we will probably be looking more at individual metabolomics to come up with treatment modalities. I mean, right today it is kind of out there, but is that the way everyone is thinking? Maybe 5 to 10 years from today, we're going to get a patient's metabolomics analyzed, proteomics and transcriptomics and everything. And for each and every person, we can come up with a very precise treatment. Is that the view now all over the world?
yeah, it is moving in that very same direction.
And the interesting thing is, again, we're going back to diabetes and diabetes being so common in India. Just yesterday, I was reading this paper so the Masala study in the US is looking at diabetes in the South Asian population. And I think the next research paper that they are going to come up with is looking at the metabolome of South Asians in general and this entire Masala cohort. That is one thing that is very interesting. So Dr. Agarwal brought up something called essential amino acids. So what does essential amino acid mean? Essential means your body cannot make it from anything else. So obviously it has to come from outside. And coming from outside, so amino acids make proteins. So coming
Yeah.
from outside means it has to come from food. And as we know, in India, protein intake is quite low. So that is again another factor. And it's not just for COVID-19, it's for all disease in general. And protein is a very important factor. So if it is essential amino acids and the body cannot make them, obviously it has to come from diet. And the other essential factor is the essential fatty acids like omega-3 fatty acids. Now, the interesting thing is there are no essential carbohydrates. So we can actually live quite well without carbohydrates, but we do need proteins and we do need fat. So next is, in the interest of time, I'm sort of rushing. Can we use lipid abnormalities as surrogate markers for immunometabolism? Because all these lipid markers are easily available in any lab.
Yeah, people have looked very closely actually at some of the lipid abnormalities and correlated with the outcome of the disease.
Mm-hmm.
However, even with this paper that we are projecting right now, it has not shown much of a difference between the subjects who had a high or a low levels of cholesterol or triglycerides or HDL. People were interested in actually LDL and HDL.
Mm-hmm.
But however, in this particular paper, there was not much of a difference. So there was a trend towards significance that patients who have got a higher triglycerides or low HDL, they are likely to have a poor outcome.
Mm-hmm.
But somehow this paper didn't actually report it that the lipid abnormalities are that abnormal and they actually are marker for prognosis.
Thank you, Dr. Agarwal. So just wanted to recap what we are talking about. So total cholesterol. Now, sometimes people associate, high cholesterol with higher risk for cardiovascular disease, and then they stop eating eggs. So in my world of nutritional medicine, we keep saying, your dietary cholesterol doesn't have anything to do with your serum cholesterol. So don't freak out with the high cholesterol and stop eating eggs. And in your world of all the autoimmune conditions, hypothyroidism is one of the big ones for high cholesterol. Then the other thing is high triglycerides is of course intimately related to insulin resistance. And normally if someone sees a high HDL and a low LDL, they would be very happy. I'm talking about regular doctors, like a family doctor would be very happy with numbers like this. But the fact is, cholesterol is so much more than just good cholesterol and bad cholesterol. There is, of course, the lipid particle number and size, and most importantly, are these oxidized or not? So my point in showing this is cholesterol is not just high or low. There are a lot of nuances to that whole entire, the cholesterol panel, because lipid panel is something that people do. And sometimes, believe it or not, I'm sure you've come across this, a lot of patients are put on a statin just because the total cholesterol is high. It's
Yes.
like, and that is not even always justified. So that is something that all my patients from India, young, old, irrespective, most of them come to me with a statin already prescribed by somebody. So this is the reason I wanted to bring it up. Dr. Misra and Dr. Gupta, would you like to add anything to this, or we can go ahead with the next slide?
No, I think next slide.
Okay. So this
Next
is
slide.
again, sorry, Dr. Agarwal, you saying something?
No, not me.
Okay. So this was, again, this is not a COVID-19 study, but what I wanted to bring up is the fuel switch, because in my world of, intermittent fasting, time restricted eating, we talk a lot about ketones, and particularly in relation to dementia, because dementia, one way of thinking of dementia, and dementia is also like an epidemic worldwide. So dementia is again a change in the fuel supply in the brain. So the question is, here we're talking about the immune system. So is it that giving ketones, or if the person achieves nutritional ketosis, is that going to change the immune response? And I'm sure no one has looked at it right now. So one is, do the endogenous ketones, nutritional ketosis, when someone goes on a very low carbohydrate diet or goes on to a fasting protocol, the body generates ketones. Is that a cleaner fuel? We look at it as a much cleaner fuel than glucose, because it works much differently. The endogenous ketones versus giving ketones as a supplement, are the two different? That is one question. And there are a lot of studies, not in COVID, but in the area of dementia research, where it shows that the two probably act a little differently. And the other question is, autophagy is something that we talk about a lot, and mTOR, mechanistic target of rapamycin, and how, autophagy is related to that. Is autophagy good for COVID-19? So there are two questions. One is the ketosis question. Is there a change of fuel required? How will it impact the immune system? And the other question is autophagy relevant? What is the relevance of autophagy in COVID-19?
Yeah. So, Latika, will you like to take up this question on ketones?
Yeah, definitely. With ketones, there are reasons to believe that, as you said, it's a clean fuel, it's more efficient. So for the same amount of ATP produced, it requires less oxygen. And a hypoxic state like COVID-19, it makes sense that keto diet, which has been explored for various other conditions, refractory epilepsy, obesity, and all that. So people are exploring. In fact, there's one very interesting study which is currently registered and ongoing, but the results are expected maybe early next year. The other reason why ketones can be useful is that they can also, particularly beta hydroxybutyrate, it can decrease inflammasome activation. It can in fact inhibit it. So the main switch there in the inflammasome inside the cell is potassium efflux. When the potassium is thrown out of the cell, then the inflammasome complex, all the components can come together and activate and produce cytokines. And beta hydroxybutyrate has this property to stop that. So an additional reason, because COVID-19 per se may not be as damaging as the inflammatory response to it is. So definitely something to look at, but whether exogenous supplementation can be that useful is again something which has not been looked at. Yes. But fasting, prolonged fasting, that is a very interesting concept, which in general can lead to complete metabolic switch, but I think we'll touch on the subsequent slides. Yes. But autophagy, yeah, autophagy, again, as we talked about the mTOR, M2, mTOR1, the mammalian target of rapamycin, and mTOR2. So autophagy can be good for COVID-19 because it will slow down metabolism. So it will also affect the immune cells in a similar way. Mm-hmm. It will cause a switch from the pro-inflammatory to the relatively anti-inflammatory state. Although it's more complex than it looks at the surface, but it is something which has potential for benefit in this disease.
Dr. Misra, would you like to add anything to that?
Not really.
Okay. Thank you. So one basic, Dr. Gupta, you brought up something very important. So we know that not from the coronavirus pandemic perspective, but in other research, they have shown that, fasting may be detrimental in a viral infection versus a bacterial infection. That is one thing that we obviously need to look at. And the other, one of the major differences that, most people who are not in the fasting, intermittent time-restricted eating world, they kind of talk about starvation and fasting in the same way. They are not the same. So starvation, as you can see, this is a picture of a famine versus a Buddhist monk, I assume, does a lot of fasting. So in fact, again, going back to the metabolomics of fasting is quite different from the metabolomics of starvation. The two are not the same. And most importantly, what we talk about when we talk about intermittent fasting and time-restricted eating is fasting is planned. A starvation like a famine was definitely not planned. And more importantly, there's a lot of stress response that goes on in a famine condition versus, a planned fasting. I know that I have access to food and the brain has a huge impact on our entire metabolism. But here I just wanted to, bring up this difference because a lot of people confuse. You'll find in papers, starvation studies show this. And I'm saying that is not a starvation study. That is a fasting study. So it is different. The two are not quite the same. And yes, as you mentioned, exogenous ketones, unless studies come out, we can't really say. But exogenous ketones are available there. And I'm quite sure there's a difference between the two. So the question is, should we recommend long fasting for someone with COVID? At this point in time, we tell people that don't get into a long fasting because we don't have the data. We don't know whether it's going to help you, it's going to harm you. But can you stick to a time-restricted eating window of maybe 12 to 14 hours? Yes, for sure. So again, maybe we'll explore that in some details later on. So some other areas that we have, in the interest of time, we haven't been able to cover is, of course, we talked about iron metabolism and hepcidin and all of that. Then dysregulation in the amino acid metabolism. Can we give exogenous amino acids, as Dr. Agarwal, you talked about? And then, of course, fatty acid metabolism, mTOR. One of the interesting, one of the papers, and all the references are there on the slides, and I'm going to upload the slides on SlideShare, because if anyone is interested to know more, some of you may feel that, we just kind of covered a little bit of everything. If you want to go and explore the details, all the references are there for you. One of the interesting thing is the cardiomyocyte metabolism that, I thought, okay, we know that the coronavirus impacts the heart. So will that, change how we recommend food? I think it will. Someone who is just recovering from a coronavirus infection versus someone who is looking at preventing it. I think that nutritional recommendation could very well be different for them. And at this point in time, this is just, expert opinions. We don't have the studies, but wouldn't that be an interesting area to explore?
Yeah,
I just
definitely.
want to put in one point here about
Yes.
fasting.
Yeah.
One needs to understand that patients with COVID-19 are extremely sick, and I have seen patients lose almost half their weight.
Yes.
It's an intensely catabolic state, so I don't think that's really going to be relevant here for patients with COVID-19.
Yes, that is the reason I brought up the exogenous ketones, because at this point in time, they're extremely sick people, and we never recommend extended fasting for someone who's so sick. So that is something very important. That is the reason whether the exogenous ketones are going to make a difference. So I think this is the summary of what all the things we talked about. And would you like to add anything more, or we'll open it for questions now?
I think we can take up questions because there is hardly 5 minutes left.
Yes. Okay, so I'm going to stop sharing the screen. And to the audience, you can type your question in the chat or in the Q&A, and our experts will be happy to respond to them. I don't see any questions yet. And I don't see any raised hands as well. Okay. And I think since we don't have any questions, thank you very much, Dr. Agarwal, Dr. Misra, Dr. Gupta, for investing your time with me here today. And this video will be uploaded. And I'm sure one of the things I wanted to do is to encourage, young researchers or would-be researchers to look at maybe research questions based on our discussion today, because immunometabolism is something which is going to have a huge impact on not just COVID-19, on a lot of other conditions as well. So I think we do have a question. Okay, I'm just reading it. Very informative talk, sir. Just can you guide, if not insulin, then can metformin be used as usual? Okay.
Yeah, so very interesting and very rightly also. I think the only drug out of all the treatments available for hyperglycemia, only metformin has shown that it has got potential to uh, do benefit. And one more benefit of metformin is that it is a blocker of mTOR pathway also.
Mm-hmm.
And there are now trials registered on clinical trials website where they are actually using drugs which block mTOR pathway for COVID-19 infection. So we will wait for the data when it will be available. But this is a sort of an area where the world is looking forward, and it makes some sense also, rational also, because mTOR pathway is playing a great role in inflammation in severe COVID-19. I think metformin should be the drug which should be carried on as far as possible.
And just to add to your response, Dr. Agarwal, if someone is on metformin to manage their diabetes, definitely they need to be on it, because having optimal blood sugar levels is extremely important to build resilience. And again, none of this recommendation is to be considered medical advice. This is for information only. Dr.
Yeah.
Gupta and Dr. Misra, would you like to add anything to that?
Yes, we definitely with the metformin, there are studies which have shown some benefit and decreased severity, decreased incidence of severe COVID in women. But we need to be mindful of the chances of lactic acidosis in the severely ill patients. So we have to look at the comorbid conditions, and maybe in very severe COVID, it may not be an option.
Mm-hmm. Thank you.
I think in those COVID who are in the ICU, it really is not feasible.
No.
Because you will need to have them on insulin. So
yeah, and if they are not on oral intake, then I don't think. Do you get a metformin injectable? I don't think so.
No,
No.
it's oral only.
It's oral only. Yeah. The other area, which of course none of us can talk about, is nutrition in the intensive care unit. I mean, some of the nutrition that I've seen, it's like a white bread sandwich. Good grief, you're going to give them more sugar. Anyway, that's a different story. Of course, most of the very seriously ill are not even able to eat orally, so they are getting IV fluids. And I think that was the only question we had. And thank you very much. Thank you, everybody. Dr. Agarwal, Dr. Gupta, and Dr. Misra for investing your time. And I am sure there will be more questions coming up in later, webinars like this. Thank you so much for your
Thank
time
you.
and your.
Thanks a lot, madam. And it's a pleasure to be on this webinar today.
Thank you, Dr. Agarwal. Okay.
Thanks.
Bye.